Hello everyone, good afternoon. Welcome to the Ensembl 91 release webinar. My name is Ben Moore. I'm one of the outreach officers who works for Ensembl. So I work on a small team that's responsible for giving workshops and courses like this one, webinars and things like that. And we also create and maintain all of the online help and documentation as well. So it falls to me to deliver this Ensembl release webinar where we quickly go through some of the new and updated data that we've included in our latest Ensembl release that was actually released on Monday this week of the 11th of December. So just a little bit of housekeeping for the webinar. Hopefully when you're logged in you should all see the go-to training box with lots of different options for the webinar. There are opportunities for you to ask questions throughout the webinar. I won't be stopping at any point to take questions, but my colleagues Erin and Astrid will be able to help you by using the chat box. So if you open the chat option, there's a small box where you can type any questions that you might have that come up throughout the course of the webinar, and they'll do their best to answer any of your questions. And if there are any still outstanding at the end, I'll do my best to answer those as well before I leave. There isn't any threading in that text box though, so if you do have any questions, then we think it's always best if you start your question with your name, so you can write @ and then your name, and then my colleagues Erin and Astrid will be able to directly respond to you by using your username as well. So it might get a bit complicated if there's lots of questions at once, but hopefully by using your name, we should be able to answer your questions thoroughly and quickly. So just a bit of background about the Ensembl project. It sort of is quite important in leading into the reason why we have this release cycle that we do. So many of you may already be familiar with Ensembl and how Ensembl works. Many of you already know that it's a genome browser, and it was first conceived of in the late 1990s, just before the publication of the first draft of the human genome sequence. So the first organism actually to be sequenced was a bacteriophage in 1977 by Fred Sanger and his team. And you can see that it was only 5 kilobases in size, which obviously is quite small by today's sequencing standards. But obviously that technology laid the foundations for more and more complex organisms to have their genome sequenced until in the early 2000s, the first draft of the human genome sequence was published. And this was 3 gigabases in size. So obviously, again, we all know that the explosion of sequencing technology has led to the cost reduction of sequencing whole genomes and also the increase in efficiency. So we have now this the birth of lots of different sequencing projects, either aimed at sequencing lots of different species from around the world, or alternatively looking at sequencing individuals within specific populations around the world as well. So with this explosion of data, what we have really is a lot of raw sequence data. And obviously, buried in this sequence data, there's lots of information, but without any context or annotation, it doesn't mean an awful lot to anybody. So this is where Ensembl as a genome browser tries to take this data, annotate it with important information, and make it accessible to scientists and users in that they might be able to then use it in their scientific research. So Ensembl tries to do this in 4 main ways. So the first is the genomic assemblies that we import. So these are publicly available genomic assemblies that we then import into Ensembl and use an automated gene annotation pipeline to predict the existence of genes along the genome. The second type of data is the variation data. So this is where we display information about both small and large-scale sequence variants with the phenotype associations as well, where they are available. The third type is the comparative genomics data that we have. So this is where we compare gene and genome sequence from species to species. So these are things like whole genome alignments and producing gene trees, which are useful in predicting homologs. And the fourth type of data is our regulatory data. So this is where we take experimental data such as DNA methylation data, ChIP-seq data from large collaborative projects, And we use that data to predict the existence of regulatory features along the genome, like promoters and enhancers and those sorts of things. So to draw these 4 types of data together, Ensembl has a number of features. So the first is, as I mentioned, the gene builds, which we have for over 100 species now, including the species in Ensembl 91. And then we take these gene builds side by side and compare them to produce our gene trees, which is, again, I mentioned, important in predicting orthologs and paralogs. For human and mouse, we also have our regulatory build. We only have the regulatory data, the prediction of promoters and enhancers for human and mouse. And also, as I mentioned before as well, the display of variation data, along with a very useful tool that we've also developed called the VEP, the Variant Effect Predictor. On top of these types of data as well, you're also able to upload your own data to view alongside all of the existing Ensembl publicly accessible data as well. And then all of this data obviously can be accessed via the web browser, which maybe you've visited before, but you can also access this data on larger scales through Biomart or through our Perl and REST APIs as well. But the best thing about all of this is that it's completely open source. So all of the data that you see in Ensembl is completely free for you to take, use, modify, and analyze, and obviously use in your own work. And we really hope that you can do that. So as I mentioned, Ensembl has over 100 genomes now. And the Ensembl project is primarily dedicated to the vertebrate species. So we try and cover the broadest taxonomic space possible while also including agriculturally, conservationally, evolutionarily important species. But obviously you can see that we have, for example, the great apes and a large number of primates, which is obviously important if we're thinking about the new Ensembl release, where we've included a lot of new primates that I'm going to speak about as well later. We also have a large number of rodents and other placental mammals, which are obviously including agriculturally important species there as well, like cow and sheep and pig. And then we also have marsupials and monotremes, so you can see how we've included evolutionarily important species in our mammalian, the mammalian species that we have available. And then we also have a large number of other birds, reptiles, fish, and amphibians as well. We do also have C. elegans, the fruit fly Drosophila melanogaster, and Saccharomyces cerevisiae as well. So these are obviously non-vertebrate species, but they're very important model organisms in biological research. So we also include those 3 species in Ensembl as well. So the reason why we're having this webinar is because we've just recently released a new version of Ensembl. So Ensembl works in a release cycle. Every 2 to 3 months, we freeze the databases and we release a new version of Ensembl. So in September of this year, we released Ensembl 90. And then since then, what we've been doing is we've been including new genome assemblies, we've been updating any underlying software, reperforming some of the analyses, adding new data and updating some of the interfaces. And then we package all of this data up and we release it in a new version of Ensembl, which is Ensembl 91 that was released this week. So the reason why we do this is that it makes everything very traceable. We know that the scientific data at the moment is very dynamic. Lots of new data is constantly being released and updated and produced. So obviously it's very important to version every release of Ensembl that we have. All the new data is all packaged up together. And you can actually view all of the archives as well. So all of the databases you can access as well, and the web archives you can look at as well for the last 2 to 3 years as well. So thinking about the release for Ensembl 91, we've included a lot of new data. So just some of the highlights that I want to speak to you about in the webinar. You can read about all of the data updates that we've included in the link at the bottom that's found on the Ensembl website. But just to quickly go through some of the important updates. Firstly, we have the incorporation of a large amount of new data for primates. So this slide is actually wrong here. I don't know why it says mouse here. So we have actually 12 new primate species, which we didn't have in Ensembl before. And we also have 6 updated primate genomes as well. So obviously we had a number of primates before, like chimpanzee, gorilla, and macaque and baboon, but we've also updated those genome assemblies as new versions of those genome assemblies have come out. For cats, we had an updated genome assembly, and then we also have updated GenCode GSet for mouse. And we also have what I wanted to mention to you as well at the very end of the webinar, a new linkage disequilibrium calculation tool, as well as updated variation and phenotype data as well. So thinking about the new primate species that we have in Ensembl, as I mentioned, we've got 12 new primate species. I'm not going to go through them all, but they're all listed here. Obviously, primates that are very important in studying human and vertebrate mammalian evolution, and also some of them are very interesting in their own right with some of their social behaviors that they display, and so obviously very interesting for biological research. So some of the primates that we've now got there in Ensembl is the bonobo, the crab-eating macaque, and Nancy Ma's night monkey as well. So you can explore all of those species that have the genome assemblies and the gene sets as well produced for them. As I mentioned, we've also updated the genome assemblies that are available for a number of different species. So we already had chimpanzee, mouse lemur, gorilla, olive baboon, and tarsier, for example. And so what we have done is we've updated the species, the genome assemblies that are available for those species, and updated the gene annotation as well for each of those species. Away from primates, we've also updated the genome assembly available for cat. So in Ensembl 90 and in previous releases, the genome assembly that we used was the Felis catus 6.2, but now we've updated to the Felis catus 8.0, which is a newer version of the cat genome assembly. And this was produced by the International Cat Genome Sequencing Consortia. So we've updated the genome assembly, and we've also produced a new gene set that's been created with the cDNA and EST sequences, but also RNA-seq data from 28 different tissues in cat, and also UniProt protein sequences as well from different related vertebrates that have been used to create the gene set as well. The final thing that I want to mention is the linkage disequilibrium calculator. So this is a new tool that we've developed that allows you to calculate the LD, the linkage disequilibrium between variants in Ensembl. So this is only available for human, and it uses data from the 1000 Genomes project. But you're actually able to use this tool to calculate linkage disequilibrium between variants within a genomic region, between a list of variants. So if you've got a list of variants that you want to calculate LD between, you can do that as well. Or you can also calculate the linkage disequilibrium for a single variant in a defined window size as well. So what I'm going to do actually is just quickly jump out of the presentation and give you a quick demonstration of how this linkage disequilibrium calculator works. So if you go to a tab in your browser and you open up ensembl.org, this is the homepage, www.ensembl.org. And then what you can do is you can click on the tools link in the blue bar at the top of the homepage. And you can actually see that we have here a list of all of the tools that are available for processing your data in Ensembl, for example. And you can see at the bottom here this new tool, the linkage disequilibrium calculator. So if I click here on the tool name, it will open up a page that allows you to submit the job that you want to the linkage disequilibrium calculator. So the first thing that you would do is to choose the calculation that you want to perform. So the first is to calculate LD in a given region, which is described with the diagram beneath as well. Or you can calculate LD for a list of variants. So if you have a list of variants V1, V2, and V4, for example, the LD will be calculated between each of the variant pairs within your list. or you can calculate LD for a given variant within a defined window size as well. So if your variant is V3, for example, and the window size is denoted by this arrow, you'll calculate the LD between V1 and V3, V2 and V3, and V3 and V4, for example. So V3 against each of the other variants within your window. So this is an example of a job that I'm quickly going to demonstrate to you now. So we're going to keep this option selected, and then you just need to give your job a name. So I'm going to call mine example. And then what you need to do is choose your 1,000 genomes population of interest. So obviously, these are all of the populations from within the final phase 3 of the 1,000 genomes project. So you just need to choose your population of interest. And then you need to include your data in the text box. So as I have mentioned above, I'm going to calculate the LD for a given variant within a defined window size. So I just need to upload a single variant into this list. So I'm going to use an RS number, RS4988235. Or alternatively, you can upload a file or provide a URL that points the LD calculator towards a text file with a list of variants, for example, in a public directory. And then you just need to choose your threshold for R squared and for D prime, as well as defining your window size. So you can see by default here we have 200,000 base pairs, which is what I'm going to use as an example, and then you can click Run. So once you've submitted your job, you'll have a page here that displays all of the different jobs that you've submitted to the Linkage disequilibrium calculator. So this page refreshes every 10 seconds, as you can see here. I've only submitted one job, and you can see here at the moment it's queued. And now, just as the page refreshed, you can see it's running. So what I'm going to do now is I'm going to click refresh again, and you can see that my job is now done, and I'm able to view the results. So now you can see a quick preview of all of the linkage disequilibrium calculations that have been performed. So each row is a single calculation. You can see variant 1 is always the variant that I chose, and then we've got variant 2, which are obviously all of the other variants in our defined window size as well, with the LD calculation scores over in the columns on the right-hand side. So this is just a preview. You're obviously able to download this data as a text file, or you can actually view the linkage disequilibrium plot as well. So if I click here, it will take you to a page of Ensembl that has the LD calculation that we defined in the web tool. And now the page is loaded, you can see we've got the LD plot for R squared and D prime, with the variant of interest here defined with this black line in the middle, RS4988235. And then for each of the variants upstream and downstream of our variant of interest, we have the either R squared or LD, the R squared or D prime scores on the graph. Okay, so just to finish up with the webinar, the last thing that I wanted to mention to you is some of the updated variation data that we also have in Ensembl. So just as a piece of background, Ensembl imports variants and variation data from a number of different databases, dbSNP, DGV archive, the GWAS catalog, and then we also import phenotype annotations from OMIM, population frequencies from the 1,000 Genomes project, and all of the sources are listed here. So obviously, when new versions of these databases are released, Ensembl will continuously import the most up-to-date version of their database with the new release. So some of the updates for Ensembl 91 include an update in the human phenotype data from a number of different databases, including the GWAS catalog, OMIM, ClinVar, UniProt, the Cosmic Gene Census, OrphanNet, MimMorbid, and DDG2P. We also have phenotype data updated for several species from Animal QTL and OMIA. as well as updated phenotype data for zebrafish from ZFin, rat from RGD, and mouse from both IMPC and MGI as well. We also have updated our variation data from DGV archive, which is a database that has information about large-scale sequence variants. So we have new and updated studies for human, cow, macaque, mouse, sheep, pig, zebrafish, and horse. And we also have updated variation data from HGMD, Cosmic, and dbSNP for human, chicken, cow, horse, mouse, pig, zebrafish, macaque, cat, and sheep as well. So obviously, if you're interested in any of these species, you can look at the species homepage in Ensembl and view the databases that have been updated, the versions for each of the different species. So just to wrap up the webinar, I wanted to point you towards some further places to go for learning about Ensembl and help and documentation. So the first thing that I wanted to mention to you is that we're actually able to host workshops at your institute. So we can come to your institute and deliver either a one-day browser course or a short 1 to 2 day REST API course. And they're aimed at different people, so either the browser course for wet lab scientists, clinicians, or the longer API courses for bioinformaticians as well. And we can teach an institute at your workshop as well. So if you email helpdesk@ensembl.org, then we can work with you to deliver an Ensembl workshop that's specifically tailored for you and the work that's being carried out at your institute. And just for further help and documentation, you can either go to the EBI Train Online resource for further information and training about Ensembl and all of the other EBI resources, or you can go to YouTube or Youku for Ensembl training videos and tutorials that we've uploaded to our channel Ensembl Help Desk. Or if you have specific questions, maybe about data that you're looking at in Ensembl, you can email the help desk using the email address helpdesk@ensembl.org, and either me or one of my colleagues will get back to you trying to help you with your query. We do also have a number of different publications. So obviously, as I mentioned, Ensembl is free for people to use in their work, and we really hope that it is useful for you. But if you do use us, then it's really important for you to cite us. It just helps us understand our audience, who's using Ensembl and what for. So we really appreciate it when people cite us in their work. It helps us to keep Ensembl relevant for people to use in the future. And then also you can keep up to date with Ensembl and obviously all of the Ensembl releases via social media. So we've got Facebook and a Twitter account where we announce things like the Ensembl releases. And we also have a blog, ensembl.info, which you can follow and obviously keep up to date with things like all of the data that we're going to be including in releases, as well as information about planned maintenance downtime and lots of other pieces of information as well. So just finally to acknowledge everyone, the list of authors here is everyone that you would have seen on the previous slide in the photo taken from our release. And then also it's important to acknowledge our funders, the Wellcome Trust, Ensembl, the NHGRI, as well as a number of other funding bodies as well that help Ensembl to maintain its standards of data and to keep the resource available for everyone to use as well. So that brings me to the end of the webinar.